A silent link: how Salmonella typhi may trigger gallbladder cancer. In the year 2020 alone, more than 84,000 people around the world lost their lives to a lesser-known but deadly disease, gallbladder cancer. Often overshadowed by more prominent cancers, gallbladder cancer, GBC, tends to fly under the radar until it's too late. But what if one of its culprits isn't just a random mutation or bad luck? What if it's a bacterium better known for causing a common tropical illness? Meet Salmonella typhi, the microbe responsible for typhoid fever, a disease millions have battled across history. But researchers have discovered that S. typhi has a much darker side. It may be playing a hidden role in the development of gallbladder cancer. More than a fever, the hidden damage of typhoid toxins. When S. typhi infects the body, it doesn't stop at just making you sick with fever and fatigue. It produces toxins, particularly a group of compounds known as nitrosochemicals that are considered carcinogenic. These substances aren't just harmful, they can actively damage the DNA in our cells, interfering with the natural cycle of cell growth and repair. Over time, this disruption can pave the way for cancer, and the danger doesn't end there. S. typhi is a biofilm builder, meaning it creates a sticky, slimy layer of protective materials called extracellular polymeric substances, EPS, around itself. This biofilm helps the bacteria survive in the gallbladder for long periods, causing chronic infections like cholecystitis, gallbladder inflammation. The longer it stays, the more toxins it can release into the surrounding tissues. The DNA saboteur, a triplet of trouble. At the microscopic level, S. typhi is armed with three powerful genes, CDTA, CDDB, and CDC. These genes work together to form what's known as a cytolethal distending toxin. The real villain here is CDDB, which has the power to break both strands of DNA inside host cells. But CDTB doesn't act alone. It binds with two other toxin proteins, PLTA and PLTB, to form a lethal trio, CDTB, PIL to PLTB. Once inside a host cell, this triplet attacks the DNA directly, inflicting breaks that can kickstart the process of cancer formation in the gallbladder, hijacking the cell's control panel. But DNA damage isn't the only way S. typhi causes trouble. The infection can also lead to dangerous changes in genes that usually keep cells in check. One of them is TP53, known as the guardian of the genome. When this tumor suppressor gene is mutated, the cell loses its ability to prevent cancer. At the same time, the CMIC gene, a proto-oncogene involved in cell growth, may become overactive. The result? Gallbladder cells start growing out of control. Adding fuel to the fire, S. typhi secretes special proteins, SOPI, SOPI2, and SOPIB, that sneak into host cells. These proteins don't just help the bacteria survive, they activate pathways like MAP kinase and ACT, which further encourage the cell to multiply uncontrollably. Slowly but surely, the infection lays the groundwork for malignancy. Bile and bacteria, a toxic mix. There's another piece of this complex puzzle. Bile. Bile is the digestive fluid produced by the liver and stored in the gallbladder. It helps break down fats, but when S. typhi infects the gallbladder, its toxic molecules mix with the bile, creating a hazardous chemical cocktail. This toxic mixture directly attacks the gallbladder's inner lining, damaging the cells and transforming the environment into one that favors cancer development. Researchers have also pointed to the Gas-Sting pathway, a molecular alarm system inside the body that responds to infections. When overactivated by chronic infection, this pathway may unintentionally contribute to DNA instability and further drive cancer formation. The bigger picture. What started as a simple fever may end with a far more serious outcome. Salmonella typhi isn't just a fleeting invader, it's a master manipulator, capable of altering DNA, hijacking cellular pathways, and creating a chemical battlefield inside the gallbladder. This growing body of research sheds light on an important connection between infectious disease and cancer, reminding us that microbes and malignancies are more closely linked than we might think. With better understanding, earlier detection, and improved public health strategies, we can begin to address this hidden threat and protect vulnerable populations from both typhoid and gallbladder cancer.